Okay, happy Pink Wednesday, everybody. Y'all haven't seen me on Wednesdays. Y'all don't know it's Pink Wednesday. It's a thing, I promise. So for today, basically any questions you have that you feel are insert things you're not clear about, we want to try and clarify them. Usually when we have office hours, we like a round robin around the room, and there because we just go in order to get to everybody. So I'm thinking what we can do is try to like zigzag from back to front. And if you don't have a question, just say skip, I'm just listening, whatever. If you have a question, please speak up and ask it. So let's start from, is this recording? It is. Let's start from the far corner here. Other things too, so if you have learning assistants in the room, say if you have a question and you just want to like have someone come like look at the paper or something, or say like, how should I word this question, just raise your hand. But just listening, let's keep it moving. Oh, okay, we'll go here. You have a question. Okay, so we're doing week 4. You said homework. And question 3. Okay, what's the question about the question? Yeah, so go ahead, read me the question stem. Actually, let me pull up. Okay, so what I'm gonna do is pull up the homework. The homework week 4. We said question 3. That's correct, right? Question 3. I'm going to read out the question stem so that everyone can hear it. If you can access it on Canvas, access it on Canvas. So we're talking about setting up a test similar to the Ames test. And this is one we talked about in office hours yesterday, and it did confuse people a little bit. So really want you to think through the genotype and phenotype of what you are beginning with. So what it says in this question is that we're setting an Ames test, and we're going to use a species of fungus. So we're going to start out with a methionine auxotroph. So we're going to call that MET delta. And it is resistant to the antibiotic kanamycin. So I'm going to put KAN resistant. This is what we are starting with. We then test the potential fungicidal compound X, and we grow it on a plate lacking methionine. So important part is that the plates themselves will have no methionine. So they will only grow if they get a reversion mutation to make their own methionine. I think that part is very, very clear to everyone. The second thing is that they have an additional gene that is not natural. They have this kanamycin resistance. So it is also possible to get a mutation where you lose this kanamycin resistance. And then having the second piece of information is what's going to help you go through the answers that are there in the question. So the 2 that are correct, it says there are fewer cells around compound X disc compared to control plate without compound X. That's one thing that we would conclude if compound X is the mutagen. And I think everyone agrees with that, yes? So on the control plate, we're putting it down so that there is no methionine. So the only way something is going to grow is if it gets a reversion mutation. And sometimes this can happen spontaneously and you get a couple of little colonies, and that's why it's not completely blank. Some random mutation that, yes. So on your control plate, remember in an Ames test, the control plate where there is no mutagen, you can still have some of the cells get a spontaneous mutation and be able to make their own methionine. That's why you see a couple of colonies sporadically. Is that part clear? And is that what was unclear to you in this question? Okay, so everyone feels good on that concept of we're starting with some type of mutant that can get a spontaneous mutation and revert back. Yes, so the clear zone is the space that's right around the disk. Everyone's clear about what the clear about. Oh, the clear zone is in an Ames test. So I want you to be very, very clear on understanding the different parts of the Ames test and what the outputs look like. You're not going to get a question that has a double mutant. Hopefully, just push you to think about the different scenarios, what an Ames test could basically give you. Okay, we're good there? Yes. Okay, so for this one, remember, think about two genes. If you add in kanamycin and they're still growing a lot, then you didn't get any mutation that disrupts kanamycin resistance. Yes, so depending on the level of mutagenesis, you could fix one or the other and not both. Hence why, because, so remember, if something on the exam is ambiguous, it's not meant to be ambiguous. Nothing's ever meant to be ambiguous on an exam question, so I'm checking your knowledge. On homeworks, discussion, I'm pushing you to think. So is it clear why the answers in the actual homework are correct based off of the ability of a mutagen to cause mutations that reverse both of these? It's focusing on both. So you can reverse one, you still get some growth, but then say you have a mutagen that reverses both, then you'll actually have what you call it, you lose that resistance, so you don't get as dense of a population of growth, because you lose that resistance. So it's by putting in either the no-methionine and then having plus or minus kanamycin that you can pick apart whether or not your mutagen also impacted the second gene. Okay, cool. Well, now it's hot. See, it's still pink, so just think one thing. Okay. So let's keep on this general back area. Do you have another question? Okay, we'll just pop up hands. We want to make sure we don't waste time. Here?
My question regarding the quiz, like in order to express the eukaryotic gene bacteria, this components and assays were expressing vector that are not found in the regular plasmid. So understanding you like because you're one of eukaryotic genes bacteria, you need a bacterial promoter and like ribosome binding sites and for the replication. And that maybe do other things as well.
I mean, that's the basics. So basically what we are asking, if we want to actually express a eukaryotic gene in bacteria, we want the gene to get made, what are some of the things that are needed in that vector? So normally you have different expression systems from eukaryotic to prokaryotic. So the whole idea is that we want to have a promoter that will turn on, that will allow the production or expression of that gene in the bacteria.
And then with replication, binding sites.
Okay, questions back here. What question was it from the?
I don't have it. It's not, it was on the quiz aid, I think.
Yes, it's definitely from lecture 8. So it's either going to be reading or homework for 8. Okay, reading quiz 8. Go here. Okay, does anyone know what is going to use or what the 3 to 5' exonuclease function is for? Proofreading. So if polymerase 1 makes a mistake, what does it do? It proofreads, it backs that thing up, and it goes opposite direction. That's the 3 to 5'. 5 to 3' is to get rid of the primer that's ahead of it as it's extending DNA. Know the difference between these two directions, what they're used for, what they do. I mean it. Okay, do we know what we mean by call one, X on the place function, 5 prime to 3 prime, 3 prime to 5 prime, 5 from the 3 prime, remove primers. 3 prime to 5 prime, proofreading. We're clear. Okay. Another question. Here. Okay, so number 7, we want to know why DNTPs and magnesium are needed for in vitro PCR. Okay, so important concept that I want everyone to know is the difference between PCR, which is in vitro, and an in vivo DNA replication. What is different between them? What is the same between them? So when we think of the whole process of DNA extension, no matter if we're doing it in vitro or in vivo, we have to be able to add on nucleotides. The whole way we add on nucleotides is by adding in these deoxy NTPs with 3 phosphates that allow nucleophilic attack. So you have to have the NTPs, both in vitro and in vivo. The magnesium is important for coordinating the actual reaction in the active site of the polymerase. Without the magnesium, you don't get the whole entire extension mechanism of polymerase 1. The polymerase 1 is not going to work. So you need the magnesium, you need the DNTPs. This is the material. This allows the actual reaction to happen. Yes. Yes, they are. You need magnesium for DNA replication, DNTPs for DNA replication. Without either of these, it won't work. So think of the magnesium as coordinating the reaction that happens in the active site of polymerase 1. Literally like holding everybody in place so that nucleophilic attack can happen and it can just kick off the phosphates. Any other questions here? Know what things you need in vitro PCR, what you need in DNA extension, what is the same, what is different. So let's go to like a lab. You will literally add the magnesium free floatings, and then because of the amino acids in the active site, the magnesiums get bound within the polymerase. So it does the function in the polymerase, but you just have to add it. Okay, any other questions while we're still here? Yes. Okay, so let's see if we can crowdsource this. If you're trying to choose between PCR and in vitro replication, what's one of the main things that allow us to choose? Size. PCR, things are going to be smaller. You'll know small by hearing things like a gene, a small piece of DNA. For in vivo, things that are large. You'll usually find things that are like greater than 5 kilobase pairs. You can say a very large gene. Or we would say things like chromosome, which is what is in that question. And so, for example, like just on exam, the concept I want you to know is that smaller things for PCR, really large things for in vivo. I won't give you something that's ambiguous in the middle. I would say a small piece of DNA or a gene. And then for in vivo, we normally say chromosome because that's a crap ton of genetic information. And then because it's, or we would say, a vector or plasmid, this is what we talked about on Tuesday. Even those are relatively large comparatively for amplifying via PCR. So we would say these things that are large, we prefer to do in vivo replication in order to amplify the amount of genetic information we have. If it's a small thing that we want to be able to amplify very quickly in vitro, we would do PCR. Usually this is for a small portion of the gene. And technically with PCR, even when we're trying to look at a gene, when we do amplification, we don't even amplify the entire gene. We pick a region of that gene so that it can actually be efficient. So small, large. Yes.
I have a question on one point. Was the one that says like, the Ames test is used to identify potential carcinogens. Compound test results in a few scattered colonies on the media, similar to the control plate with no this. What does this imply? So I think this implies that compound is not mutagenic, so there was no increase in reversion rate. I was trying to understand that. But my only understanding is the few colonies that grew to contain a version mutation that allowed to grow on a media with that machine, which kind of contradicts the first one. I was
Okay, so actually, we're gonna back up. Go back to the Ames test for a quick second. Reset your minds. On the control plate of an Ames test, we start off with an auxotrope that technically should not be able to grow because it doesn't have the nutrient it needs. But experimentally, you will always find a couple like stray colonies throughout the plate. And the question is, what are these colonies? Why are they able to grow? So what happens is that sometimes you can spontaneously get a mutation that causes that reversion. And this is representative of spontaneous mutations that can occur. So your negative plate in an Ames test is never going to be completely clear, ever. And the reason being is that they use the auxotrope that they know is susceptible to this actual spontaneous reversion. Okay, clear?
the alternative could be that it's also a big, it's not mutagenic at all, you get no effect, and another part could be like, oh, there's still a little bit of new effects.
So the only way you know that the mutagen has an effect is if you see the growth in a circle around the mutagen that you put in. When it's scattered like this, that's nothing to do with the mutagen.
It could just be random.
It's random chance reversions. I saw the hand over here.
Thank you.
A spontaneous mutation. Yes, that's fine. Keep asking. Okay, so now we're on to the topic of homologous recombination versus non-homologous end joining. And the whole idea here is that we have designed a plasmid that has a gene, this gene doesn't really matter, and we want to use this plasmid to knock out something that's in the genome. In order to knock out this gene, the whole idea is that we're going to utilize the fact that the cell can do homologous recombination, meaning that if there are homologous sequences surrounding the gene that match whatever this gene is up here, then it'll utilize HR, homologous recombination, to basically let these two genes switch places. So that part we're all very, very clear on. So now the question is asking, what could potentially happen if you have a cell that has the capability to do both of these, right? So several options. Number one, the cell can take up the plasmid and express the genes, and it doesn't get incorporated or knocked out the way it should get knocked out. Because this is not a 100% efficient reaction. Remember, none of this is ever 100% efficient. Option number two has the ability to do the non-homologous recombination, and this works perfectly. You knock out your gene and you get what you intended. Number three, because it has the ability to do non-homologous end joining, depending on if there's damage, where it is in the cell cycle, you could potentially try to incorporate some of this DNA anywhere else in the genome. All it has to do is process it and then ligate the blend ends together. So those are the three possible options. So for example, if I did this and then I decided to have a cell that for some reason had issues with non-homologous end joining, no more end joining, yeah. then we wouldn't have the outcome of getting it basically incorporated anywhere in the genome. That would no longer be an option. If we thought of the converse, where we said, this one can only do non-homologous end joining. It doesn't have what it needs to do homologous recombination. This entire approach wouldn't work and we just wouldn't get the knockout. We can incorporate the plasmid, you probably get inserted somewhere randomly in the genome, but we wouldn't get this directed, very specific knockout. And in labs, we actually use this whole idea, the way that the cells do non-homologous end joining, and that's how we make these transgenic mice. We'll just randomly insert genes into genomes of places and hope it works out well. Okay, questions on this?
Yeah, well, so it was like, I think actually the next question got more into it, where it's like, where are the sequences, I guess, like, what is required for the selection marker specifically to place the gene of interest? And instead of mentioning the sequences surrounding the gene of interest itself, not the selection marker. So does that have to go with something like that diagram, where it's like the two crosses or?
Yes,
something
yes.
like that? I don't know how the drawing looks like, but.
Okay, let's do this. I'm drawing, this is my vector. This is the DNA. This is the genome. This is DNA. We technically care about this gene and what we're trying to knock out. But the way to get this to happen is that the parts that have to match and be homologous are what's surrounding it, or what we call flanking it. This has to match. So this part has to equal each other. And then this has to match. And then that's how you're going to get the homologous recombination. And remember, if you think about how homologous recombination works, the whole idea is that it uses a template to basically make sure we don't lose genetic information. And so you end up getting like little breaks here, invasion of the strand, and because like they're aligning, then it ends up doing that weird switch. It's a really interesting mechanism. Yes. Okay, so it goes, what questions again? Are we on? I found no questions. It's homework. We for question 6. Okay, so the question is why the sequences surrounding the selection marker will be placed around the gene of interest. What is the activation marker? What does that mean? Oh, it's kind of worded weird. So I agree that the wording is strange and it's not a selection marker will be placed. Yeah. I don't like the wording of this either. I'm going to rewrite it. So based off how we're interpreting it, like this is true. You need these sequences to match.
On the gene of interest. Both the genetic and selection.
It will be gene of interest and selection marker.
Okay.
And I think the wording just wasn't clear enough. Is everyone clear on this concept?
Yeah.
I'll make sure the concept is clear. And like I said, if you run into something on the test, it is ambiguous. Don't stress out. Make a note of it and talk to me after the test. But the point is in the trick, y'all, is to test your knowledge. And if it's ambiguous, then it's not doing that. Yes. Exactly. So no matter what, this doesn't impact whether or not you can transform that plasmid into the cell.
Right.
Yeah, it's impacting how it gets incorporated into the genome if it does. Yes. Exactly.
I have a question on book number 5. Where it
Okay,
asks,
pause. Are we done with this? HR and ESJ? Okay, not done. No, they don't have to be the same. Yes, can be random. Okay, any other questions on homologous recombination, NEHJ? Okay, we have a whole cluster happening here. Do you want to go check what questions they have? Okay, cool. Yeah,
okay, sorry.
so the selection marker is whatever gene that we're trying to transform into the cell. That lets us know that transformation would have worked. That's the selection marker. Okay, so clear there, yes? Okay, you had a question here?
Yeah, just, on number 5, on the book was, why is it carrying a single stranded break more than a double strand break? It turns out, like, it doesn't require like another component to repair, and it doesn't result in the loss of nucleotides, but doesn't technically homologous combination also share genetic info and prevents the loss of nucleotides, or not? Maybe we divide too much. Reason that it's so big loss in a homologous combination is just the copy is the same.
Okay, so you're thinking really, really hard. So a single strand break. So we have DNA. And I got a little break. For the most part, if it's only a break, what do I need to do to fix it? Just ligase it. Low energy input. If it was somehow missing a couple of nucleotides, what do I got to do to fix it? Just DNA polymerase and ligase. So single strand breaks are relatively straightforward to fix. They're not a huge issue. You don't have to go back and like delete stuff additionally in order to fix it. Is that clear? Okay, next question. Here in the back.
Yeah. Okay, so you need to I haven't spent time in looking at the details, like to guide the option, the entire sequence, and if they were to ask you what kind of sequence what is that? Is that?
You write the reverse complement.
So,
So anytime I'm asking, so if you're paying a company to sequence their DNA, do you want what the thing, the DDMTPs was, or do you want to know the sequence of your DNA? You want the sequence of your DNA. So when I ask you on the exam, can you tell me what's the sequence of the DNA that was sent out for sequencing? I want that sequence. So just remember, you have your whole, so your gel. There's A, B, or C. Remember, this was 3', this was 5'. We end up making some type of whole sequence based off of the gel. Don't give me this sequence. Give me the reverse complement. Yeah, and there should be an example in your notes. Use your notes to be like, okay, this is how I do this, and give me the reverse complement. Okay, yes. Yes, that's a template string. Your reverse complement for Sanger sequencing is the template string. Okay, any other questions on Sanger sequencing? The most recent material, stuff we did yesterday. Go to the midst of the whispering. It's okay, just do like that. Okay, so our whisper groups, have we got it figured out? We want to ask the question out loud. Y'all comfortable? Yeah, okay, y'all. Okay, we got thumbs up. Okay, yeah, question here. Will the gel always be this orientation? Yes or no? Yeah, so the always this orientation, because gels always run, let's call it, the little pieces towards the bottom. So it's always in this orientation. You don't have to like, it's really funny because I tend to actually, for anything else like 50-50, like left and right, like I just mix them up all the time. So things like this, I just tend to write out and have it somewhere accessible. I don't know, my daughter knows left and right better than me. It's a thing. Okay, any other, yes. Okay, so any other questions on Sanger sequencing? Okay, we'll keep it moving. Go ahead. Okay, so what we're asking here in the question stem is that essentially you cause mutations to cells and then some of them die. And that's what you're looking for, and that's look all that applied question is any type of process that the cell needs to live. That's what you're looking for in that answer. So is it clear how we're getting to that logic of, okay, I had my cells, presence of UV light we know causes mutations and then they died. So we're looking for mutations that will specifically cause a cell to die. What is a process necessary for life? That's what you're looking for. Is that clarified? Okay, and just to double check, that was practice exam question number 4. Yes. So what type of point mutation was it? Can you read the whole answer? Okay, I'm gonna open up because I couldn't hear over the door. You said this is, right now I'm in homework. I'm gonna go back. I'll open up practice exam. A, B, C, D, E, F, G, H, there we go. It's the same question, yes? Okay. And the back of the canvas is annoying. Click so many things. Click, click, click. Okay. And number. Okay. I guess it's not number 7 for me. Number 4. Okay. Okay, he said okay, so we're on this question. I found it. And the question you had, okay, a DNA that represents long coding RNA had a point mutation. Okay, so we talked a little bit about like different types of DNA and RNA and what they do. The important part that makes that answer not correct is that it's in long non-coding RNA. So we're not interrupting the production of any genes for the cell. It's not coding. That's the important part. Is everyone clear on why the answer isn't correct? So it's not the point mutation part, it's where the mutation occurs. And that's what this question is asking. Yes. It would be a possible answer. That would be very ambiguous because what kind of gene is it in? Or is it deleterious or not? Is a mutation that results in actual change in the protein? So I can give you more information, but point mutation in a coding region would be a potential answer. Yes. Well, you don't know. It could result in cell death, depending on where that point mutation is. It's because where you have the point mutation, it's in non-coding. Basically, essentially, the code is for a long non-coding RNA. It's not coding for a gene. And knowing some of these differences, you're going to get into different types of RNA and translation next week. Yeah, it's happening. Any other questions on this particular question? Yeah, so if you're having issues with knowing like which mutations are what, if you go back to your reading list where it talks about mutations, I would literally, for those ones, go back to the textbook, maybe make yourself a little cheat sheet and list of missense mutation, nonsense mutation, truncation, what does it result in? Like have that clear and like accessible to you. They're kind of important things to know. Yes. No, in this context, no. They talk all about DNA synthesis, but there are plenty of pathways in cells that if you got rid of them, say it was a histidine synthesis pathway, and this histidine is essential for life. I would spell it out for you. So there are multiple pathways that are essential for cells to live, and get mutations in them, of course, the cells would die. The DNA synthesis, though, is one that all of you should know. Yes. So once the mutation is literally part of the genome, it's a mutation. So if you think about examples in class where we have a patient that would say patient X has this mutation, and then they get this type of disease or pathogenesis. It's because that mutation can't be fixed. It's already part of the genetic information of that organism or individual. Yeah, once the truncation is encoded, it's encoded. I mean, technically, this is, I'm going to do the exam. Of course, people are trying to find ways to do gene therapy where they will introduce new genetic information and things like that and try to repair like cells, like using stem cells and differentiating them and giving them to people. So the potential is there, we just can't do it right now. Okay, other questions, back to the exam. Okay, so I think the question is, where's Frank coming from? There, you said mismatch repair pathway. Why do we need to know which side is the daughter strand? Okay. In mismatch repair, essentially you have, let's just say this is a C, C, A, T, G. And then polymerase just made a mistake, it effed up, right? So it's supposed to be G, G, instead of putting a T, it's just stuck in another G, and then you have your A, and then you have your C. The big issue is that you're gonna get some structural abnormality here because we're not hydrogen bonding the right way. So your cell knows, oh, I gotta fix this, but both of these are supposed to be in DNA. They're both DNA nucleotides that are normally part of your genetic code. So the only way to figure out which one to cut out and replace is by figuring out which one is the new strand, which one's the old strand. And that's why the cell has to do that. Is that clear? Okay, cool. Yes. Okay, yep, and you know this too. So we're gonna now move on to helicase, and one of the experiments that was set up so we can understand what direction the helicase moves. Shift it down. This board is like making us hard to read. Okay, so you will run into this, and you already have run into it, hopefully multiple times, discussion, practice exam. Direction of helicase is going to be determined by the strand that it is attached to. That's number one. And then so number two, experimental design was essentially set up to figure out what direction this helicase can go. So what scientists did was they used radio-labeled probes. One is a 796 mer, one is 722. And the whole idea is that if this helicase went from 5' to 3', it would knock off this little piece of DNA. And if it knocked it off, when we ran it on a gel, it should appear towards the bottom because it's small. Yes, question. Yes. Experimentally only. So when you design this experiment and you go like synthesize a piece of DNA, it has directionality. You know which side is 5', you know which side is 3' because of your free 3' OH group. Yeah. So we're dictating what type of helicase it is based off of what it's bound to. So remember when it's separating a strand, just for a second, think of a second DNA strand right up here. And this is 3' and this is 5'. From our perspective, it's opening the two pieces of DNA. And technically, the strand it's attached to is moving 5' to 3'. But the other side that is complementary, it's moving 3' to 5'. So the way that we define it is the one that the helicase is actually attached to. That's what we mean by that. Okay. Now back to the experimental example. We want to determine which way this helicase is going. So once we actually set up this experiment, we run out the products on a gel. We always expect to see some at the very top because no reaction is 100% efficient. So say for a tube that only has a 796 mer, if we knocked off the 796 mer and we saw a little bit of that signal towards the bottom, then we'd expect that the helicase is going from 3' to 5' because it knocked off the one on this end of the DNA. Does that make sense? Okay, so for example, if my gel instead had something where there was signal towards the top, so basically that's indicating the long piece of DNA that still has a radio-labeled piece of DNA attached. And then instead of seeing a signal on the gel for 96 mer at the bottom, I only see signal at the bottom for the 722 mer. What does that tell me about the direction of the helicase and why? Go from 5 to 3, and why is that? Piece at the bottom, you came this way, boop, knocked it off, and now you can see that small piece at the bottom of the gel. If for some reason you saw nothing at the bottom of the gel, what would that tell me about this experiment? Helicase didn't knock anything off, we didn't have it in the first place, it's a helicase that was a dud, it didn't work. Exactly, what if I showed you a gel where there's 2 of these? Both got knocked off. Bidirectional helicase, it has the ability to go whatever direction it feels like. Yes. So either its helicase didn't move, because this means nothing was knocked off, or maybe you forgot to add helicase. This is what the gel would look like. You would have no signal, the helicase didn't knock anything off. Okay, so I came back here. No, it's one that would just have the ability to go whichever direction. So it wouldn't have like, I'm just gonna get a friend and let's go opposite. It just has the ability to go either way.
We have like a billion of these things, and go on here, if you pack them, we can go on, happen, we can go on,
Okay, question here.
There's like some of the after vision exams, and you're like, oh, what side of the abilities go? Let's say we put 5, 3 and 3 and 5, and then 5 direction, how do we look at the options, will we get it wrong?
No, if you set it on both, then it would go both, that'd be correct. And so if you said that it went both and only goes one way, then that wouldn't be okay. So if it goes both, make sure you select both. Don't select only one direction, even though technically it goes one of those directions. Okay, any other questions about this helicase experiment? Is everybody gonna get it right tomorrow? Yes. Yes, we are. Okay. Next question. Yes.
I think just to clarify it positive, so that the market can make a student. I don't know if the, what my thoughts of it is.
Okay, we're gonna talk about negative selection versus positive selection. Either way, they are still selection. The easiest way to think about negative selection is that we are actively killing cells that don't take up the plasmid. So for example, if a plasmid with ampicillin resistance, and then the cells, we're going to add in ampicillin. The cells that have the plasmid are going to grow, but we're killing the cells that are going to die. So what we are adding to the tissue media or to the culture is killing off things without the plasmid. Negative selection. Positive selection is going to be the opposite. So say, for example, we want to do one of those like knockouts of the histidine gene in order to create an oxytroph, right? So let's say this is my plasmid. I want to swap it out with the genome that has the histidine gene. After I swap it out, the ones that got my plasmid are now actually histidine delta. They are oxytroph. So I am going to add in histidine to ensure that these grow. So the thing that is supporting the growth of what is transformed is positive selection. Of course, in this scenario, you need something else to kill off the rest. But positive and negative selection is just what you're putting in order to ensure that you're only growing the cells that you want. If you use hand in hand, most people don't even really think about positive versus negative. They just make sure we are selected. Yes. Okay, I'm gonna double check where you are in the reading, because it should be that you're killing off the things that you don't want to kill off the things that conduct the plasmid. So we just want to make sure we clarify, so I'll double check in the text what might be misread, but we never want to kill the things that we're trying to select for. We want to kill off what we don't want, whatever didn't take up the plasma. Yes,
So when why did you
oh, so what I did here was I did a swap of some other gene I cared about, and then I just knocked out the histidine gene within the genome. So I don't want us to get too caught up in the difference between positive selection and negative selection. What's most important is understanding how selection works. And know that researchers use both positive and negative selection to get to the same outcome. Histidine oxytryph. Yeah. So this one by itself isn't selecting, you'll need something else in addition. And ideally, this gene that we knocked in would be an additional selection. Yes,
You said that we wouldn't want to kill the cell, but we wouldn't want to actively kill the cells that not transplant.
yes,
I don't know if I'm reading the problem wrong, but the lemon four, the E problem is about. He was actually said sometimes the negative selection, we should get to the standard positive selection marker. And it's going to look for active and negative selection marker. I don't know. Sometimes like non-defined. Second marker, add a second marker in the knockout business. Success that it's inserted into the genome, so it's all spelled check. When you play it on the selected.
okay, so let's go here, we're going to send homework 4.
Yeah.
Number 8. I do want to make sure it's clarified, so don't feel confused, because feeling confused is not good. Okay, so talking about homologous recombination, basically this process, we want to confirm selection markers integrated for place in genome. One must know selection markers using the standard positive selection, which one would be active negative selection marker. Okay, so basically what they did is what we normally don't want. This is just another level of double checking. So, so they're like, okay, can we selectively kill it? Is what they asked. Yeah. Yes. Right. And it is opposite. And honestly, like in the lab, I wouldn't take this approach. So I think for like future homework, I'll do a different question. But remember on the homeworks, I'm trying to push you to think, it should not be ambiguous on the exam. So does the concept of negative selection make sense? Negative selection is adding something to kill. Right? You're selectively killing something. So in that homework question, that's what was done. But I would never try to actively kill the thing. Yeah. I wouldn't approach an experiment that way. Yes.
So if you like adding any antibiotic would be negative and adding any like amino acid that the cell can be positive. So
Yeah, that's the easy way to kind of give yourself a dichotomy. Like trying to kill as opposed to like trying to make live. Yes. Yeah, controls. Okay, so moving on from selection, make sure you understand selection. Like I said, for the exam, do not get caught up on positive versus negative selection. It's a good thing to know, but do not get caught up spending your time studying it. We are now moving on to controls. Exactly. When you're thinking of a negative control, think about what is the baseline? What is the standard? What are we measuring against to find a change? That's what you think of when you think negative control. Negative control will change with different experiments, but if it's genetic, your control is going to be the wild type, right? It's the normal way that the genes are expressed. And then you see that in the XP experiment that we have with the UV cross-linking and stuff going on there, and the whole like NER repair pathway. Positive control is something is basically a experimental condition where you know what the outcome is supposed to be. This outcome has already been tested and validated in another space, and this allows you to know that any outcome you find in any other sample is true and valid because the assay is working the way it's supposed to work. Think of this positive control as the way to know that your experiment works appropriately. That's what the positive control is. Are those two things clear? Okay, good. Know them. Okay, so we're like at 12:50, so people who have classes at 1 o'clock, if they need to like leave or like take places, feel free to go. We won't be disrupting us, I promise. Okay, yes, question here. Think of the negative control as your baseline or your standard. What we're going to compare our experimental against to see if there is a change. Yeah, you always got to have a negative control. Most of the time you need a positive control, but very few times, sometimes positive controls don't exist. Yes. It's usually wild type in a genetic study. Wild type, negative control. Any other questions on positive and negative controls? Important concept, not just for the exam, but rest of your life, for anything science. No positive, negative control. Yes. So we're asking about why is there a clear zone? Why is there a place where a mutagen basically doesn't allow cells to grow? Yes. So think about like a double-edged sword. The mutagen at a very high concentration will cause so much DNA damage that you have mutations and pathways that cause the cell to die. Too much mutagenesis is bad, cells will die. As you go out from this dip, you finally get to a concentration that is low enough where you are getting the reversion mutation, but you're not killing off the cells. Then eventually it gets so diluted that you don't get an effect. Correct. Okay, cool. Any other questions since we're back to Ames test? Yeah. Both are correct. So the mutagen is increasing the amount of mutations, so more chances of getting a reversion mutation. And then because it is causing those mutations, you will see this pattern of a ring growth around that disc. Yes. So for the point mutation, it's not a deletion. For this oxytrope, because it's fixable or revertible, is essentially, say, instead of like a G to C, you have an A to T, and you cause a mutation, you end up like reverting it back through repair mechanisms. So it's reverting it back to the ability to grow. Because remember, these plates, you're starting off with the oxytrope, it can't grow without the histidine. Then you don't add any histidine to this plate. So no histidine is possible. The only way it's going to be able to grow is if it gets a reversion mutation and it's able to grow again. So the whole idea is that this has to be a point mutation because we have to be able to cause a mutation, have repair pathways turn on, and somehow, or like miss or not be able to repair it, and then you end up getting a mutation that reverts back. But then if you have a deletion, you've lost genetic material, you can't fix a deletion. A deletion is a deletion. So yeah, so if say we start off with oxytrope and we decide to use an oxytrope that was caused by a deletion, if you did an Ames test, you'd always get a negative result. Because you can't fix that mutation, no growth. Correct. Yes.
So let's say your sample is not, it's technically right, yeah, because plate that looks similar to those scattered cells are also one that like spontaneously.
Yes. Okay, so I'm in here. We know a reverse mutation occurred because cells are growing. The only way the cells grow is because the mutation reversed. That's basically it. Whether it's like a mutagen or not, any colony you see that grows on the plate is because of a reverse mutation. Yes. It always starts off with the knockout. We're looking for induction of growth. And the whole reason, so the particular oxytrope that we use in the Ames test is really susceptible to that reverse mutation. Like why it is, we don't know, but because it's susceptible, it makes it easy to use as a model organism to test whether or not we can see if this is mutagenic. Go ahead, no, go ahead, you're good. Yep, we can do that too. Okay, my question back. Yes, so your negative control for the Ames test is just the oxytrope plate with no mutagen stuck in the middle. So remember baseline, what we're going to compare to see if there's a difference. So this would be, if you want a positive control for this experiment, it's going to be a no mutagen, something we know causes mutations. That would be a positive control.
Okay.
Yes. These scattered colonies. Yes. Other questions on Ames test? Yes.
You can hear the color, right? How many genetic compounds.
Yeah, you could. And so it's all going to be concentration dependent. So you can tell how mutagenic it is based off how big that clear zone is. And then you compare all the different mutagens using the same concentration. And say, how big is the clear zone? How mutagenic is it? The bigger that clear zone, probably the more mutagenic it is. Oh, that's a great question. Sorry, it's an exam question next year. Oh no, erase that. Not an exam question. Okay, kidding. Any other questions on Ames test? Okay, any other questions in general? Yes. Yeah, so the figure in textbook sucks. So what I want to say about mismatch repair, the individual proteins, the mu S, mu L, like that stuff is important. What I want you to know is that this whole entire repair mechanism is important for repairing errors of DNA polymerase. So one thing was wrong. There's a little bit of bulge in the DNA because they're mismatched. Essentially, you have to cut out multiple nucleotides or to fix it, then you fill it in and you ligate it. And the other important point is that you basically have to discriminate which strand has mutation, which one doesn't. That differs by prokaryotic versus eukaryotic. We use phenomethylation in prokaryotes and eukaryotes. They say that we use mix. Technically, scientists don't even really know how this works, but this is like the theory that's ongoing. Yes. Yes.
Okay, so we're trying to find
Exactly. Yes. Any other questions on mismatch repair and what you need to know about it? Yeah, so hemimethylated means one side is methylated. In prokaryotes, we're saying that there's a delayed methylation of DNA. The parent strand is going to be fully methylated. The new strand or daughter strand, not so much. So they're looking for differences. And basically it realizes that the strand that has less methylation is going to be the one that I have to fix. That's what this whole system is basically discriminating between. Yes. Nope, not gonna be asked that. Just, oh, okay. Yes.
and then try to run this. But that is basically are the when you do more than one.
Yeah, it's just telling us what, it's just one old, one new.
Oh,
The old one's already methylated, the new one still has to get methylated.
so this is when it's activated, that means publish nine with one.
Mhm. Exactly,
The parent methylated, sorry.
yes, exactly. Any other questions on mismatch the pair pathway? Okay, next question, or any other questions, clarifications?
Is this more about the Illumina sequencing, that we talked about that?
No, we did not talk about Illumina in the lecture. If it's not covered in lecture, it will not be on the exam. But there are some resources, and I think I dropped a video on campus about Illumina sequencing. It's basically how we get from Sager sequencing, which was the baseline, to being able to do one reaction where we have more efficient Sager sequencing and then now next generation sequencing. So it's a cool thing to watch based on fluorescence. It allows us to do really, really high throughput. You're not going to be quizzed on it, but I want you to have the resource there. If you go to a laboratory doing sequencing, it'll help you. The Sager sequencing helps you understand the basis of how this works, and then I would just really dig in if you're in a sequencing lab. Okay, other questions? Yes. Oh, blunt ends. I heard float ends. I'm like, I don't know what that is. Okay, we're gonna go over blunt ends versus sticky ends. Blunt. Sticky. Okay, blunt end versus sticky end. The whole idea is that we have endonucleases nucleases that can cut DNA or nucleotides based off of the sequence. And when cloning, we usually prefer sticky ends. We usually prefer sticky ends. Most gaps you can find like that. Because it is more precise. We know exactly what the ends look like and then where they're going to join. With blunt ends, you can literally join any two pieces of DNA together because they're cut flat. It'd be like, oh, I don't have one marker. It's such a good example. Oh, that's so sad. I don't know. I don't have the thing I was going to use for an example. But for example, the cap can only fit on this marker in one way. Boom, boom, boom. And then so it has directionality. With the blunt ends, it can essentially, as long as you have like the 5' and the 3' end, they can just get put together by ligase. So it's not sequence specific once it's been processed this way, and this is why it's not preferred. Is that clear? Okay. Yes. They still have directionality, so it still have to be like 5', 3', 5', 3', but it could be like any particular sequence. So hopefully that wasn't confusing the way I said that. There's a question here. Exactly, precision and stability. I like that. That's a really good way to summarize it. Sticky ends allows to have precision in our coding. Okay, any other questions? Yes. Any utility to larger versus smaller? Not necessarily so. All sticky ends from like, so the cool thing about these endonucleases is that they're all like naturally occurring and they recognize sites themselves. And for the most part, the range of the size is actually relatively narrow of how long we can actually make these ends. But these aren't actually controlled by like experimentalists. I would assume that a longer sticky end would give you even more specificity, and then of course you have more hydrogen bonding, but practically there's really no difference. Yes. Anybody else have a question? No, okay, these are different things. They should be annotated if used on a question. But HU is hydroxyurea. And then H. pylori is a bacteria. Is everyone aware of the example, I guess, figure experiment that this is referring to? This is it, okay. So let's make sure you know the difference. The hydroxyurea, this is a positive control that we know is supposed to induce chromosomal breaks and instability. And then H. pylori was suspected to cause chromosomal destabilization or, breaks or mutations, whatever. And so these were the experimental ones. As you increase the concentration or MOI, multiplicity of infection, how much bacteria you started with. If you increase how much bacteria you started with, you cause more and more DNA damage. And that's what that experiment was showing. So no difference between these two things, they're not the same. Exactly. Specific on concentration because we're testing it. It's the bacteria, and then by having this, hence the Ames test, you know how we do dilutions? When you show that something is dose dependent, you increase the confidence that the outcome is what you say it is. No, because that's what we're testing. That's the question we have. Positive control is this molecule that we know causes chromosomal damage.
So the question we have is, whether increasing the concentration of bacteria or
Mhm. Okay, other questions? Yes.
Discussion with the person about ideas and. I'm saying we're advocating for, it said that you found that we have no ideas. So we're. We can make I get that adding pyrophosphate kind of breaks down like pyrophosphate, so it was but for is that based on, it was adding more ACP like to react as a reactant. Would it push like would it push it for a force of like the values in the work because different products we study the way we like.
Right.
So adding more reactant like this is enough.
So the whole idea here is asking if you're going to troubleshoot to figure out what could have went wrong, what are things to try? So something to try is adding more ATP, maybe it was too low. It might be the ATP wasn't a problem, right? But logically, based off of knowing the mechanism, this is something that you could try. Same thing with pyrophosphatase. But your understanding of chemistry is spot on. Any other questions, clarifications? Oh, yeah, that I've debated whether to not to do this experiment multiple times. It's actually pretty involved. If you want to stay after and go through it, we can. It's not on the exam. So she's asking about, there's a figure in the textbook that goes over how they figured out whether or not the beta plant was important for processivity. Oh my gosh, English. And I think it's a really cool, elegant experiment. We just don't go over it in class. Yes. Yep, that is why. Okay, any other questions? Here. Okay, so in the mechanism, we know that we need ATP in order to add on the, or for the DNA ligase to form that intermediate or adduct, whatever, in order to do the nucleophilic attack. So an idea is that if you're doing a reaction and DNA ligase is not working, what are potential things I could try to make DNA ligase work? Potential thing you could do is think, well, maybe I didn't have enough ATP. Let me add ATP to ensure that this important step in the mechanism is being fulfilled. So that's why you add the ATP. May or may not. So don't overthink it too much. Think about, once again, the question stem, what am I asking? If something went wrong, what could I try to fix it? I'm not asking what would actually fix it. what would I try to fix it based off of understanding the mechanism? If you understand the mechanism of DNA ligase, you know it means ATP, you know it involves like kicking off the pyrophosphate, and that if you have too much pyrophosphate, you can do like reverse reaction, so let's try pyrophosphatase. And that's why those two answers are correct for the discussion worksheet. Any other questions? Okay, if we don't have any questions on content, any questions on logistics? Exam's gonna be in person, same thing as on a week and a half ago, so fast. Same thing as like a week and a half ago. I can't map anything. Okay, if you have any issues, normally with like plugging in your laptop, whatever, and you need like a place to plug in, I suggest you take it in the alternative room. I think there's still space. I haven't checked it in the last hour because I've been here. So just sign up, make sure you can access a plug. If any of you have DC accommodations, I double check, make sure everything's in place. It should be good to go. I will have paper copies of the exams in case like the Wi-Fi just dips on you. Because it can happen because, you know, PSLH. But other than that, like the questions you are asking, you are digging deep, you're understanding the concepts, like you will do well. Be confident in your answers. On the select all that apply, I'm going to reiterate it as much as I can. If you are not 100% certain of an answer, don't click it. It's not worth it to click an answer that is wrong. It is a penalty. It's better to miss something that's right than to click something that is wrong. Yes, any other questions?
For Emily's class, is this structured the same way, is it down to like open notes? Like,
Yep. Yeah, so me and Link, we're a team, we're like parents, right? Tag team, we do this. So we construct the whole entire course together. We go over the material that we want students to learn together. We make sure like it gels and it makes sense. Like he literally takes my practice exams. And so we work on this class together. So just because I'm not lecturing doesn't mean that I'm not involved. So still here, same structure, same like point totals, come in, get your check-ins, just check it the same way too. Only differences are philosophy on exams. So personally, I find his material more difficult. I find his exams easy. So if you keep studying the way you're studying, understanding concepts, understanding the why, like you're all gonna crush this class. Okay, so I'll see y'all tomorrow. Oh, you can come up, okay. Is it content or is it like?
It's like a quick read.
Okay, rest of y'all free, you can go ahead and ask what you wanna hear, what you have to ask.
Lecture 7, you said like they were both reasons causing double stranded breaks. We need to know like how those things are causing double stranded, or those are just how.
Yeah, and you can also look at it in your notes, like,
Yeah.
Pinko, if you can see it in your notes very quickly, you don't need to memorize it.
Yeah.
Yes. Okay, I'm gonna stop this recording and I'm gonna come to you, because I can't hear.